Good afternoon, and welcome to the USDA Human Nutrition Research Center on Aging's Fall Monday Seminar Series. Before introducing today's speaker, let me remind you that your questions can be added to the Q&A feature at any time during the presentation. No need to wait until the end of the presentation. So it's my great pleasure today to introduce our speaker, Professor Esther Aarts. Professor Aarts is a principal investigator at the Donders Institute for Brain, Cognition, and Behavior, and leads the Food and Cognition Research Group at the Center for Cognitive Neuroimaging. Her research focuses on functional neuroimaging to examine the neural mechanisms of eating behavior and effects of foods on brain functioning, including the roles of the gut microbiome and immune system, which she will discuss today in her presentation titled Nutrition and Cognitive Neuroimaging, Understanding the Gut Immune Brain Axis in Aging. And with that, let me turn the microphone over to Professor Aarts.
Thanks for the introduction, and thanks, Paul, and thanks for having me. It's my great pleasure to be here and talk to you today about my field in food and cognition and my research. And my research career actually started with investigating dopamine. And dopamine is a neurotransmitter in the brain, which is projecting a lot to the subcortical regions in the brain, the basal ganglia or striatum, and the prefrontal cortex in the brain. And we need these regions to control our actions, also in the face of reward. And recent theories really put dopamine into the role of the coding the expected value of actions and cognitive control. And the cognitive control part is really coming a lot from our lab. And it's basically coding whether an action or a cognitive effortful process is worth the effort. And why is this interesting for aging? Is because aging is accompanied by a decline in dopamine signaling and also by impairments in reward processing like reward anticipation and also cognitive control like working memory. And interestingly, it's not just the ability to perform cognitive control, but also a reduced willingness to expend the effort for cognitive control in the view of a certain reward. And in my work as a postdoctoral fellow with Roshan Coles and also with Mark Desposito at UC Berkeley, I've investigated the role of dopamine and the effects of reward anticipation on cognitive control. And we did this with PET and medication studies in ADHD and Parkinson's disease. And in all of these studies, we found that really whether the dopamine, or whether an anticipated reward has a beneficial or a detrimental effect on cognitive control really depends on the level of dopamine that someone has. So we see this with PET, whether there's a detrimental effect in high dopamine subjects. We see it with ADHD, differential effect versus controls, and we see it in Parkinson's disease. So this is a bit about my background. Now, one study that we did in aging was a cognitive control task to see whether a certain cognition is worth the effort. And this is a working memory task, an MBEC task, where depending on the figure, you would have to say whether it's an easy, like a one back, which is easy. You see the M here, you have to go one back, and you would say yes, it's the same, or no, it's not identical. Or it can be very difficult if it's a three back here on the right side. You have to go three back to see to answer whether it's identical, yes or no. So this is really a difference in load, and the higher the load, the more cognitively effortful. And here we show the role of dopamine. And by the way, what the important thing is that participants then later, after doing this task, have to choose whether they would like to do some trials again for a certain amount of money. So for instance, the circle is the easy option. They could choose, do you want to do the easy option for a small amount of money, or do you want to do a hard option, like a three back, for a larger amount of money. So here you can see how people value cognitive control and whether they think it's worth the effort to perform a difficult trial again. And with physical effort, it's already shown that dopamine has a role in this. With cognitive effort, there was not much evidence yet. And we wanted to show this by looking at tyrosine, which is actually a nutritional intervention because tyrosine is an amino acid which we can find in food and which is a precursor of dopamine. And we did a double-blind, placebo-controlled crossover design with quite a large amount of tyrosine, I have to admit, which is not something you would easily get from your daily diet. And we compared tyrosine versus placebo in older adults. And what we found in this study is that this nutritional dopamine precursor administration in healthy older adults really enhanced the willingness to invest cognitive effort for reward. So what was found before for physical effort, we now also find for cognitive effort and in older adults. So now let me come more to the core of this presentation, which is the body-brain interactions. Because why is dopamine so interesting? I think it's really interesting because dopamine levels might be really particularly sensitive for the metabolic demands that come from the peripheral body to shape this effort allocation or our motivational priorities. And for instance, the dopamine cells in the midbrain have all these receptors for peptide hormones like insulin, for instance, so that signal hunger or satiety. But dopamine is also very much influenced by inflammation, which I will show you later on, and the gut microbiome. And this reasoning came from Michael Treadway and colleagues, and I'll walk you a bit through this picture. And why is this important? Because again, inflammation and gut microbiome are also altered in aging, as you might well know. So it's not only dopamine processing that's altered, but there's also these bodily processes that can affect dopamine that are altered in aging. So if we look at these pictures, there are several inflammatory pathways, basically, that can influence dopamine processing. So there's more a pathogen-like pathway via NF-kappa B, and there's a cellular stress pathway via the inflammasome. So these inflammatory pathways lead to an increased production of pro-inflammatory cytokines, and they also, via glycolysis, lead to an increased energy consumption. And with an acute infection, this is really like 60% increase in energy consumption. So you can imagine that your motivational priorities then also shift. But there's also lots of disorders where we see this chronic low-grade inflammation, where there's even a 30% increase still in energy consumption. And as you might know, there's also this inflammaging. So in aging, there's also aging is also accompanied often by an increase in low-grade inflammation. So these pro-inflammatory cytokines can also cross the blood-brain barrier, partly, and then activate the microglia to produce even more pro-inflammatory cytokines. And this activates, for instance, reactive oxygen species. And here is the link with dopamine, because reactive oxygen species influences a coenzyme, which is or cofactor, which is BH4, which is very important for the production of dopamine. So with reactive oxygen species going up, there's a decrease in dopamine synthesis. And there's also effects on dopamine reuptake, for instance. Well, to study these effects of infection or inflammation, we have also done some studies in young, healthy adults. And one is it's quite severe. It's an LPS challenge. So here we again wanted to look at cognitive effort, and now with an arithmetic task, which could be they just have to add up the numbers, which could be easy if it has 2 zeros, and or it could 2 ones, or it could be a harder task. And again, there's a training phase, and then again a decision phase where participants have to say, do you want to do the hard option or an easy option? And here they would get a hard option, which is all different numbers than ones, which is harder than the easy option with 2 ones. So here we again look at accept or reject choices. And in this study, there was in young adults, we gave an inflammatory challenge. So this is actually at the Radboud University Medical Center. It was a study for the immune effects of LPS, and LPS is an endotoxin, and you'll get immediately an increase in cytokines. So this is a really severe challenge. People are in the medium care unit, so they're well controlled, and they're ill for a couple of hours. And then it's over, and then after 1 day, people can go home again. So the LPS is injected intravenously, and we did behavioral testing at several time points. And what we see first is the cytokines, that these pro-inflammatory cytokines really go up, they really raise, especially 2 hours after the LPS administration. And then at 3 time points, we had them do this cognitively effortful task, but especially decisions whether they wanted to do the hard or low, medium, or high effort option. And especially in medium effort, we see when the cytokines peak, that also their acceptance rates go down. So the participants under LPS challenge choose less of the effortful options. Low effort and high effort, we didn't see these effects because we think low effort was too easy and high effort was actually too difficult. So there was already a low acceptance rate anyway there. So this study showed that if we have acute inflammation in young healthy adults, that this also really reduces the willingness to invest cognitive effort. It does not affect reward sensitivity, or it does, so it did also not affect the ability to perform the task. So they could still do the arithmetics, they just were choosing not to do it. So this is again showing an effect on the cognitive side, which was something that was already shown on the physical effort side. Now, in a current ongoing study, we looked in older cognitively healthy adults. This is just an observational study, and this is part of an intervention study, or 2 interventions, which I will show you later at the end of this presentation. But we looked already at some of the baseline data, and there we were assessing an index of neuroinflammation. So with the peripheral inflammation possibly going up in aging, we can also look at the effects on neuroinflammation by using MRS, magnetic resonance spectroscopy. And often we look at myo-inositol, which are these 2 little peaks in the spectrum. And we choose here 1 voxel in the dorsolateral prefrontal cortex. And what we see here is that this is myo-inositol in the DLPFC on the x-axis, and the y-axis there's a processing speed. And we see that with myo-inositol increasing, which is an index of activated microglia, so activated brain immune cells, we see that processing speed is going down. So in this really preliminary finding, we see that this MRI-based neuroinflammation index in older adults predicts reduced processing speed. Here we didn't look at whether it's the ability or really the motivation or the effort, but this is something for future studies. So what I've shown you so far is that dopamine is important for cognitive control and also cognitive effort allocation, and is affected in aging, and that peripheral inflammation is also really crucial for cognitive effort allocation, and this is likely going via dopamine, and animal studies have also shown this more directly. And increases in neuroinflammation relate, at least observationally, to decrease in processing speed in aging. So now, to go back to this picture, there was also the word dysbiosis here. And with dysbiosis, we mean that it's a kind of ill-defined term, but it's generally thought that this means an imbalance in the gut microbiome. And I think many of you know a lot already about the gut microbiome. So just the human microbiota in general, we have trillions in our body. They weigh approximately 0.2 kilograms. They're mostly in the colon and the large intestines, and we have quite some species. And there's, in contrast to our human genetics, there's really a lot of differences between individuals. And the interesting thing is that the gut microbiota are really influenced by diet and also by stress. And there's important links with the immune system in the gut. So looking at the gut microbiome more specifically, I think many of you know that it's very important for absorbing nutrients, synthesizing vitamins, protecting against infection by maintaining the epithelial barrier. It's critical for the development of the HPA stress axis and the immune system. And it also produces hormones as well as neurotransmitters and their precursors. And I come back to this later because this is really interesting for the gut-brain axis. So gut dysbiosis, a kind of a gut microbiome out of balance, for instance, decreased diversity, or at least differences between a matched healthy control group, is really observed in many disorders, and also in neurological and psychiatric disorders. So this was really a beginning of this gut-brain field, is that since we could more easily sequence the gut microbiota from the stool samples, we could see that there were differences between people with a neurological or psychiatric disease and controls. And I'll also show you an example of work that we did in that field. But this is mostly based on observational studies. Well, another really kickstart of this field were the studies of germ-free mice. And I'm myself not a rodent or an animal researcher, but these mice, they are born sterile with a cesarean. They're raised sterile, and you see all kind of differences. And they were initially studied for the differences in digestion, in the immune system, but then they also saw differences in behavior. For instance, reduced anxiety-like behavior, but also cognitive deficits, social behavior deficits, increased stereotyped and repetitive behavior, and all kind of other differences when they were looking more closely at the brain, like in neuroprotection, also in stress hormone signaling and neural function. So as said, I'm not an animal researcher myself, but I must say that these germ-free mice, they have a short lifespan, so they are also they are not representative of a normal mouse without any germs. They have lots of problems, but it was really a beginning of the field that these mice did not only show differences in the immune system and digestion, but also on behavior. So that was really an important link towards that there is perhaps an effect of the gut microbiome on the brain and behavior. So there's lots of more work since then, and what I just showed is that the immune system might have an effect on the brain via increasing perhaps neuroinflammation, at least also via affecting motivational priorities. And of course, the gut microbiome has a really important role in maintaining and developing the immune system. There's also other routes like circulating metabolites from the gut microbiome. so that might cross the blood-brain barrier. And there's, of course, the vagus nerve that is connecting the enteric nervous system with the nervous system of the brain. So with the gut microbiome producing all of these metabolites that can have an effect, again, via the immune system or via neurotransmitters and their precursors, we were wondering whether we can use functional neuroimaging like fMRI to show the relation between the gut microbiome and the brain. And I'll show you now some study results in young adults, and those are the basis of the present studies we're currently running on aging. So first we had a study about ADHD. So we were interested in whether gut microbiome differences relate to ADHD. And we were especially focused on the gut microbiome potential to produce precursors that relate to a brain dopamine. Dopamine itself does not cross the blood-brain barrier, like serotonin does not cross the blood-brain barrier, but the precursors like phenylalanine and tyrosine, which I just mentioned in this administration study, they can cross the blood-brain barrier, and they are also produced by the gut microbiome. So we were interested whether these differences were perhaps specifically in the monoamine precursor pathway. So with the monoamines, I mean dopamine, serotonin, and noradrenaline, because these are the ones that also have been clearly related to ADHD, and ADHD is also, for instance, treated with dopamine and noradrenaline reuptake inhibitor, like Ritalin, methylphenidate. So we had participants hand in stool samples, and these were adolescents and young adults, and also matched control subjects, age and gender matched. And what we then get is these kind of beautiful graphs, and where we see in different levels of taxa, from species to genus to family, etc. We see differences where they whether a species or genus is increased in ADHD or increased in controls. And I must say this is a 16S rRNA sequencing, so actually the gene, the species level is not so accurately, we could not estimate this so accurately, so it's better to look at the genus level. What you see here is that we see, for instance, that Bifidobacterium was increased in ADHD, which is interesting because Bifidobacterium is generally thought to be a beneficial bacterium, which you often also find in probiotics, for instance. But I must say that when we correct for all the comparisons that you make for all of these different taxa, we really didn't find anything to survive multiple comparison correction. But we had a more targeted approach. So based on this 16S rRNA sequencing, we did a prediction analysis with PyCrust. So it's not actually shotgun metagenomics that we looked at all the genes, but we did a prediction of all these genera, what kind of genes they would have, and then what they were able to, what kind of proteins they were able to synthesize. And this way we had 17 candidate pathways in predicted tryptophan, phenylalanine, and tyrosine synthesis. So tryptophan is a precursor of serotonin, and phenylalanine and tyrosine are precursors of dopamine and noradrenaline. So we had these 17 pathways, so we also had a much less of a multiple comparison problem. When we then corrected for multiple comparisons, we did find one predicted microbiome function to be increased in ADHD, which is a CDT, which is an enzyme involved in phenylalanine synthesis. And interestingly, this predicted functionality in CDT on the y-axis was very strongly related to the relative abundance of the Bifidobacterium genus, which is also what we already found to be subthreshold increased in ADHD. So there was a strong relation there. And the interesting part, which was actually my part in the whole study, was to see whether those differences in the microbial dopamine precursor synthesis relate now to differences in brain responses for reward anticipation. So this relates to motivation. So this is the task people, as actually unfortunately only a subset of our sample, mostly non-ADHD participants. They also went into the scanner and they were doing a task in the scanner, and they were seeing these screens with where they saw whether they could earn a high or a low reward, and then if they saw the square, they had to press as quickly as possible a button, and once they were in time, they actually got the reward. So this is really a motivational task. And why do we choose this task is that in ADHD, here you see a meta-analysis, there is really hypoactivation of a region in the striatum. So this is a slice through the brain like this, where you see the striatum is with the caudate and the putamen. It's really in all of these studies underactivated in ADHD than in controls. So we wanted to see whether any gut microbiome potential related to dopamine synthesis could perhaps have a relationship with this. So we did this, we looked at this task. We had 50 versus 1 cent trials, and we compared many trials, so they could earn a bit of money, and we would see whether for more high versus low reward cues, whether there was a first just increased activity in the striatum, and as expected, indeed, the striatum is really activated for this task, which is well known. So the task worked, which is nice. And then we looked at this specific CDT enzyme, which was related to phenylalanine synthesis, which is a precursor of dopamine and also noradrenaline, but there's a rate-limiting step in between. So we looked at this bacterial gene functionality and in interaction with the reward effect in the brain. And then we still see an effect in the striatum. And plotting this effect, it was a negative relation. So the microbial phenylalanine synthesis related to decreased reward anticipation. So this was across ADHD, but there were only 6 participants with ADHD. The rest was were healthy controls. And what we see here is that with an increase in this synthesis of this phenylalanine-related enzyme, microbial phenylalanine-related enzyme, which was increased in ADHD, you see a decreased reward anticipation, which is also reward anticipation is typically decreased in ADHD. Well, this was really an exploratory observational study, but it linked microbial function with brain function. And this is one of the first that did this. And so we found that this microbial potential for dopamine precursor production related to striatal reward anticipation in young adults. But there were some limitations to the study, and I want to show you this today because that is also the limitations that we often see currently still in the gut-brain field. So this was an exploratory study, it was an observational study, and it had a rather small sample size of in the end only 28 participants who had both a stool sample and a brain scan. We only looked at predicted bacterial function based on 16S rRNA sequencing. The direction of the effects were unexpected. So Bifidobacterium is generally thought to be a beneficial bacterium, and also phenylalanine production was increased, but the dopamine-related function in the brain, which is reward anticipation, was actually decreased with increased microbial phenylalanine production. And when we did a larger study, we did not replicate this. We did replicate one of the findings, the Coppercoccus genus, but the effect of Bifidobacterium could no longer be replicated because another sequencing primer was used. And I'm telling you this because this is really what the field is suffering from, because there's lots of different primers used and different processing of the fecal material and different sequencing different bioinformatic pipelines, which all seem to have effects on the data, which is why what we see in gut-brain studies, there's not a lot of replication currently. And this is also another example of this, where we very recently did a systematic review of correlational studies of the relation between the gut microbiome composition, so looking at the taxa again, like the genera, and functional brain connectivity, so looking at resting state networks in the brain, where the participant is lying in a scanner just for 10 minutes, and we look at how the different networks in the brain intrinsically connect. You see here the different resting state networks, like the default mode network, salience network, frontoparietal network. And you see here the different genera. And really, we could not see any really consistent results from all of these studies. One solution might be perhaps to look at a more multivariate approach, because we have the brain with all its voxels for activity, but also all of its networks, if we look at this resting state and networks. And we have, of course, all the taxa in the microbiome with the microbiome composition, which are both really multivariate measures. So in another fairly recent study, we did a linked independent component analysis to take into account the multivariate nature of both modalities in microbiome and in brain. And this was work by my colleague Niels Kohn. And there you can look at how the different subjects load on the different components. And I'll show you what we got. So these are the different components that we got out of the Leica, where you see both a microbiome component, which is in green, and a component of a resting state network, which is either a frontoparietal right network or frontoparietal left, an executive control network or default mode network. And I'll explain like one component. For instance, this component 1 on the top, you see that Prevotella 9 genus was more abundant and the Blabsia genus was less abundant, with increasing between subjects functional connectivity of the core hubs of the default mode network and the executive control network. And we have these pictures to show that we really see a mapping in the brain that really makes sense in terms of these networks. So, well, this is in a way interesting, because overall we see that certain microbial genera are associated with the normal expression of all of the 4 canonical resting state networks and their natural variation between healthy young adults. However, it also left us quite puzzled because we don't really know what this means. So we see these multivariate associations, but we don't really know what this means. So what we're currently doing is really, can we replicate this in a larger sample? And also, can we associate these components actually to differences in behavior? So, so far, I wanted to show you that the gut microbiome composition can be linked to functional brain connectivity, but that this has led to quite some inconsistent results in the literature, and that maybe multivariate association methods might be more promising, but also they need replication. This was all observational, and now I think it's interesting to look more into intervention studies. One of those interventions could be probiotics, because if we talk about these animal studies like the germ-free mice, but also with antibiotics or microbiome or fecal transplants, we see these behavioral changes. And some of these studies also show that these changes can be reversed by probiotics. So here I mean live bacteria that might have health benefits. And well, the idea is that probiotics could have an effect by, for instance, protecting the epithelial barrier, which then there would be less immune activation, and this could in the end have an effect on neuroinflammation, on decreasing neuroinflammation, or at least certain degrees of neuroinflammation. And this would then also have effects on dopamine processing via the process that I showed in the beginning. One paradigm that we used in one of our probiotic studies was an acute stress paradigm. And why was that? Well, here we see stress on the left side, and acute stress is interesting because it increases the intestinal permeability to LPS. So these are parts of the cell walls of gram-negative bacteria. This is also the substance that was injected in these young healthy volunteers, where we see this really change of motivational priorities. And it also indeed increases levels of interleukin-6, this pro-inflammatory cytokine. And interestingly, cognitively, it can detrimentally affect working memory performance, specifically a type of working memory like the backwards digit span, which asks for some modulation, a bit like the MBAC, which I showed before. And it's known to be affected, this type of performance, by doping medication in Parkinson's disease. So we really think this is also a kind of a dopaminergic type of cognition. Well, in turn, probiotic supplementation versus placebo in a meta-analysis has shown to lead to a reduction in inflammatory markers, improved cognitive function in Alzheimer's disease and cognitive impairment, and also a reduction in symptoms of depression and anxiety. But here, these meta-analysis show it really depends on the type of questionnaire, type of depression questionnaire, whether you see an effect. But at least these meta-analyses so far show that it might be really interesting to look a bit further into these type of interventions also in aging. Well, we first did an intervention in young adults, and we wanted to see whether a probiotic really develops to protect the intestinal barrier, at least in vitro, would affect brain and cognition, and whether this effect would depend on stress. This was indeed in young adults, and two groups of young adults received either probiotics or placebo for 4 weeks, and they went into the scanner before and after those 4 weeks, and they also did a stress test, which I'll explain a bit in a few slides. So first, in the scanner, they got 3 different tasks: an emotion reactivity task, which really beautifully activated the amygdala as we expected, an emotion regulation task that activated more the medial part of the brain, which is also really as expected, and a cognitive control task, which activates more really these frontal and striatal regions in the brain, which is the Stroop task, which is also all very much as expected, also the behavior. However, looking at the effects of probiotics versus placebo, we did not see any effect whatsoever on any of the 3 tasks. Also not on any of the questionnaires that we had. And here I must say we had a very healthy young student population, which did not show any signs of depressive moods or anything. So our first and main conclusion of this intervention in the paper was, in a healthy population, we do not expect any effects, or we do not find any effects of probiotics versus placebo. Now, we also had a stress test. This was after scanning. The participants were in the lab, and there was a more grumpy researcher that I didn't meet before, and that told them to hold their hand in a bucket of ice water. So it was quite a painful stressor, but it was only lasting for 3 minutes, although they didn't know that it would last only for 3 minutes. And before and after the stressor, the participants did a backwards digit span. So they had to repeat a string of numbers, which was getting longer and longer, so more difficult, and they had to repeat it backwards. So quite a difficult task. And what we see here is that only in the probiotics groups there was a beneficial effect of the intervention on this stress-related working memory. And in the placebo group, we did not see this. So we concluded that probiotics resulted in a buffer to stress-induced disruptions in working memory. And this is interesting because this is also, working memory is also typically a feature that is affected in aging. Now, we did this test after scanning, but we also had of the same subjects, of course, their MRI data, their fMRI data. So we looked at this cognitive control task again and how they activated the brain, because this kind of Stroop cognitive control task, where they have to indicate the color of the word and not be distracted by the meaning of the words. This is really highly related to working memory performance. So we looked at within the groups who was showing the biggest benefit on this working memory task and whether this related to the brain functioning. And only in the probiotics group, we saw that the ones who were, where the probiotics had a beneficial effect on the stress-related working memory, they also needed less recruitment of their prefrontal cortex. And we did not see any of this effect in the placebo group in gray, which was basically a flat line. And we looked at all the regions of the brain that show the frontal regions that show the main task effects but also after correcting for multiple comparisons, you still see the difference between the probiotics and the placebo group. So this probiotics effect was related to altered prefrontal cortex recruitment. And then later we finally got funding for the sequencing with 16S rRNA sequencing of the stool samples. And there we see there was a difference in 8 genera for probiotics after versus the placebo group. And only for one of them, we did see, after correcting for the 8 comparisons, we see that was when participants showed probiotic-induced increase in a Ruminococcaceae genus. There was also a more beneficial effect on this working memory task after stress. And we did not see any of this relationship in the placebo group. So here we concluded that in a healthy young sample, probiotics only had a beneficial effect on cognitive control when preceded by acute stress, and that this related to probiotic-induced changes in prefrontal cortex responses and in relative abundance of a Ruminococcaceae genus. And this fits well with the literature, where also meta-analysis show that in healthy individuals, there's no evidence that probiotics has an effect on cognition. So whereas there was this effect in Alzheimer's disease and MCI, but it was in terms of cognition and beneficial effects of probiotics, but there's no such evidence for healthy aging or cognition in general in healthy young adults. Well, this effect in the Ruminococcaceae genus is perhaps via producing anti-inflammatory intestinal barrier protecting short-chain fatty acids, but we did not measure this. But this could be, this is of course an interesting avenue to continue to see what was exactly the effect. Was it an anti-inflammatory effect, and was it perhaps mediated by short-chain fatty acids? So this brings me to the current studies we're running in aging. So one of these studies is in 60 to 75-year-olds who are at risk of cognitive decline based on their lifestyle factors. For instance, if they have overweight or obesity, or if or diabetes type 2, or very little physical exercise. And here we look at the effect of a nutritional supplement, which is focused on the gut, and I'll get back to what this is in the next slide. And we look at the effects on the brain, but via the bodies, via the gut microbiome, and we specifically focus on the short-chain fatty acids here. And in the brain, we focus on the prefrontal cortex and the hippocampus in several scans, but the primary outcomes are functional MRI during a working memory task. So this is in it's a more or less an exploratory study in two smaller groups where we look at a certain vitamins versus placebo. And the vitamins are special in that sense that they are coded that they are only released in the colon. So normally the vitamins are, of course, taken up by the small intestines, but we know that, for instance, for the production of short-chain fatty acids, vitamins are needed as cofactor. So we also need some vitamins to reach the colon there for the bacteria there to produce short-chain fatty acids. And if we eat very fiber-rich vegetables or fruits, these vitamins might reach the colon. But here we wanted to test this effect of colon-delivered vitamins more directly with a specifically developed supplement by DSM. And we are interested whether this has effects versus a placebo after a 6-week intervention. And our primary outcomes are functional MRI and performance during the MBAC working memory task. And that is on the brain level, and on the gut level, we are very much interested in fecal short-chain fatty acids. This is together with the Wageningen University. But I've talked a bit about nutrition now, but the gut microbiome is influenced by many other lifestyle factors. Sleep has an influence, besides diet, also physical activity, medication, stress. So we're running in parallel another study in a similar group. It's not the identical group, but it's the same inclusion criteria: 60 to 75 years old adults who are at risk of cognitive decline based on lifestyle factors. And here we do a multi-domain lifestyle intervention. And we would like to see whether this has an effect on brain functioning, again, via bodily factors like in the gut microbiome and in the immune system. And here were various outcome measures on brain health. And the main outcome measures here is, again, working memory during fMRI and also brain perfusion with arterial spin labeling. So the intervention is here in also not a very big group because it's not an effectivity study. In the same study, MOCIA, we, in the same overall research program that I lead, we also have a large effectivity study with similar lifestyle domains where we have more than 1,200 Dutch older adults going through these lifestyle domains for 2 years. And this is really with a neurocognitive test battery as the main outcome measure. But in my lab, we run a smaller version of the study, only for 6 months, and with a smaller sample to really look at more the biological measures in the gut microbiome, the immune system, and of course, brain functioning. And with the gut microbiome and the immune measures, again, being done with Wageningen University. So the lifestyle intervention includes diet, which is a Mediterranean type diet. It's exercise, relaxation, sleep, and cognitive training, strategy training. And our main outcomes are fMRI during working memory task and cerebral blood flow, and also then the blood inflammation markers and gut microbiome composition. So I hope to have told you that dopaminergic brain functioning is related to the functioning related to motivation and cognitive control can be modulated by nutritional, inflammatory, and gut microbiome factors. And that the future nutritional and lifestyle interventions likely run in this overall research program, MOCIA, should reveal whether intervention induces changes in these functions in microbes and in inflammatory markers can positively affect brain functioning in aging. And perhaps also the motivation to sustain such an effortful lifestyle change. So I'd like to thank my collaborators at the Radboud University Medical Center, who are much more on the gut microbiome side of the work presented. and my group at the Donders Center for Cognitive Neuroimaging. And thank you for listening.
Thank you very much for that very interesting presentation. I'm just waiting to see. So far, we don't have any questions. I'm just curious, and some of the, I mean, in terms of the work you've done looking at the stress, the impacts of stress on the probiotics. Have there been any type of work like that done in older individuals, or looking at other forms of stress that we might see changes in cognitive functioning?
Not that I'm really aware of. I think people assume that an older individual might already be, at some point, already have some stress, like some inflammation going on, some low-grade inflammation, which is why there's not an extra stress challenge there. But I think it would be actually very relevant, because I think, if anything, a stress challenge would have larger effects even in aging. So it could be very relevant to look into this group as well. I know there were plans of colleagues of mine to look into it, but in the end, I'm not aware of any of these studies. But it would be very interesting to look further into this.
I still don't see any questions. If people have questions, please get them into the Q&A box quickly. Let's see. I'll wait another. Oh, here we go. All right. So a question from Shandong Wang, and he says dopamine can be generated by the gut microbiota, and 50% of dopamine in our body is stored in the gut. And dopamine affects our GI motility. In your ADHD study, can you determine a functional dopamine system in the gut due to altered gut microbiota rather than the brain?
Yeah, I think this is a very relevant question. So you hear a lot about this, also about serotonin and dopamine, and like most of our serotonin is in the gut, or at least indeed half of dopamine is in the gut. I always find this interesting, because dopamine and serotonin would not cross the blood-brain barrier, but their precursors would. That's why we looked at the precursors and not dopamine directly itself. However, of course, there's also the vagus nerve, and these, what we know as neurotransmitters, might also affect the vagus nerve and still affect brain functioning, although they would not cross the blood-brain barrier. So, but how this would exactly work, I think, especially in humans, we don't know yet. And the interesting thing is, with gut motility, indeed, dopamine and I think also serotonin, they have indeed a big role in gut motility. We also take gut transit time into account in our studies, for instance, by having the participants eat blue muffins, and we can see how fast the blue color goes through the digestion. And we take this into account to make more sense of our gut microbiome findings, because they are really dependent on gut motility. And we do see that transit time in very preliminary studies seem to also relate to aging, and relate perhaps even to cognition. So the transit time is important, and the transit time is probably influenced by dopamine in the gut. So this could in itself already have an effect, perhaps also on brain functioning. But how this exactly works, that's something really I would like to find out in future work, but I don't know yet.
Another question just came in from Ed Salzman, and he asked, can you elaborate on how your vitamin supplements were designed to be released in the colon and not earlier in the gut?
Yeah, I think it's a pH dependent layer there. So depending on the different pH, the layer dissolves, and then so with the pH going up, the layer dissolves and then it's released at the right place depending on the pH.
And I have a question from Parveen Bawa, who asked, can taking probiotics, if not needed, hurt? How does one know one needs probiotics in an aged people?
So far, it's not known that probiotics would have a detrimental effect. At least we show that when there's also, and the meta-analysis also show, in healthy aging without any cognitive problems, so in cognitively healthy aging, there does not seem to be a benefit to take probiotics. So I can only talk about cognition and the brain, by the way. So that does not seem to be its benefit, but it also doesn't seem to hurt, just perhaps for your wallet, because it's expensive to take probiotics if it doesn't have an effect. But it could also have an effect on your digestion, and so this could also be beneficial for people's well-being, of course. And there also come back to the transit time. For instance, in Parkinson's disease, one of the very earliest symptoms of Parkinson's disease is constipation, and that's something happening like decades sometimes before the diagnosis. So this transit time is something that's really important, and if probiotics could help with that, maybe this could be beneficial also for brain health later on. But we simply don't know yet, and we also have to be careful in Parkinson's disease itself, because the lactobacilli, which are often in probiotics, and this is one of the only things I can now think of that might be detrimental, is that the lactobacilli have certain enzymes that degrade levodopa, which is a precursor of dopamine often given to Parkinson's disease patients. And lactobacilli are often part of these multi-species probiotics. So if you give Parkinson's disease patients probiotics, they might need more of their levodopa medication, which is of course something you would not want. So that's the only detrimental effect I can think of for people using medication, and these certain bacteria being able to degrade the medication before it's taken up into the bloodstream.
The next question comes in from Larissa Constanel, and she asks, how big are the inter-individual differences in dopamine levels in older adults?
That's a good question. So it depends how you measure it. So it's often measured with PET imaging, and then you can look at the transporters or the receptors or the dopamine synthesis. And then even in synthesis, there's quite some work at the Bill Jacobs lab in Berkeley, showing that synthesis might even be increased in aging as maybe a compensatory mechanism. So with all the different elements of the dopamine system, it's hard to say what the levels are. So it's in animal studies, it's easier to just measure the dopamine with microdialysis or something. But with human studies, you always measure just one element of the dopamine system. So it's hard to say, but there are definitely individual differences that have also been related to cognitive impairments. So there are differences, and they are interesting in light of cognition, but I cannot quantify this really, sorry.
Okay, well, having no other questions in the queue right now, and having come to the top of the hour, again, first of all, I'm looking forward very much to your findings of your ongoing studies, and I want to thank you very much for your presentation today.
Thanks again for having me. It was my pleasure. Thanks.
Thank you, Dr. Aarts. We really appreciate this. And if there's nothing else, have a good evening.
Yes, thank you so much.
Bye. Thank you very much.
Bye. Bye.